Welcome back to this next video in the series of lectures on the cell cycle. If you remember, in the last video I told you that the E to F transcription factor is released from the retinoblastoma protein by the action of the G1 CDK, and by G1 CDK I mean the complex of the cyclin D and the CDK4. The G1 CDK was actually phosphorylating the retinoblastoma protein and was converting the retinoblastoma from the hypophosphorylated form into the hyperphosphorylated form. And when the retinoblastoma protein was converted into the hyperphosphorylated form, it was not able to interact with the E to F transcription factor, and the E to F transcription factor was released. Now, once the E to F transcription factors have been released, what are the functions of the E to F transcription factor in the cell cycle? Now, this E to F transcription factor is going to do a lot of things. We will be mainly focusing on the main events that are happening in the cell cycle because of the E to F transcription factor. Now, the first thing it do is that it is going to increase the level of cyclin in the cell, which is known as the cyclin E. Now, this cyclin E, when its concentration is increased, as you all know that cyclins are the regulatory subunits of some kind of the cyclin-dependent kinase. So in this particular case, the cyclin E is actually working as a regulatory subunit for another cyclin known as the cyclin-dependent kinase 2. Now that means that cyclin E is going to bind to the CDK2. Now if you look at the complex of the CDK2 and the E, this particular complex is known as the G1-S CDK. Now we call this as the G1-S CDK because this complex actually helps you in moving from the G1 into the S phase of the cell cycle. Now when this G1-S CDK complex that is formed, what are the functions of this G1-S CDK? If you remember, I've told you that at the origin of replications you have got this protein complex known as the origin recognition complexes, and these origin recognition complexes are actually working as a docking site for a lot of proteins. Now when the origin recognition complex is present at the origin of replication, during the G1 phase of the cell cycle, other proteins, the first protein, the CDC6, were actually going to bind to the origin recognition complex. After that, the CDT1 protein was binding to the origin recognition complex, and then the MCM ring was actually going to bind to the origin recognition complex. And the complex of the origin recognition complex, the CDC6, the CDT1, and the MCM ring, that was known as the pre-replicative complex. For short, we were abbreviating that as the pre-RC. Now, when the G1-S CDK is get activated, it is going to bind to add extra protein to this pre-replicative complex to you can say to prepare the origin of replication for origin firing. Now a lot of proteins, they are added to this pre-replicative complex. I am just going to give you a single example, that the CDC45 protein that is going to add into the pre-replicative complex, and that will be converted into the pre-initiation complex. And this pre-initiation complex is actually going for the origin firing. That means that you are starting the replication of the DNA. This pre-initiation complex is actually working as a platform on which the DNA polymerase and the associated proteins can bind, and they can actually go for the replication of the DNA. Another important function of the CDK2 E complex and the G1-S CDK is that it is going to phosphorylate a protein known as the P220, and by phosphorylation of this protein you are actually promoting the transcription of the histone genes. You all know that these histone genes are required for the proper packaging of the genetic material. You are making a nucleosome out of it. You are actually using a histone octamer, 147 base pair of the DNA, to make a nucleosome, and you are actually packaging that DNA. Now, if you are starting the replication of the DNA, you are actually making a new copy of the genetic material in the cell. That means you need histone proteins in a very large quantity so that you can actually pack the new copy of the genetic material properly. So another important function of the G1-S CDK will be to promote the transcription of the histone genes. One of the important things that you need during the cell cycle is that you should duplicate your centrosomes. Now the complex of the CDK2E or the G1-S CDK is also important for the initiation of the centrosome duplication, because if you are not able to duplicate your centrosome, you cannot get a bipolar attachment of the sister chromatids, and you cannot move them into the opposite poles, thereby you cannot make two normal cells. So these were just some of the important functions that the G1-S CDK that was doing. Now, once you have made the pre-initiation complex, you have fired the origin. That means you are in the S phase of the cell cycle. Now, when you are in the S phase of the cell cycle, what the E2F transcription factor is going to do now is to increase the level of another cyclin known as the cyclin A. Now this cyclin A and the cyclin E binds to the CDK2. Both of them have the ability to bind to the cyclin-dependent kinase 2. Now if the cyclin E is there and it can bind to the CDK2, if the cyclin A is there that can bind to the CDK2, what are you going to do then? Now, once you are in the S phase of the cell cycle, you have fired the origin of replication. That actually lead to decrease the level of the cyclin E in the cell and increase the level of the cyclin A in the cell. Now that means if you are decreasing the level of the cyclin E in the cell and you are increasing the level of the cyclin A in the cell, the CDK2 is going to bind to the cyclin A. Now, how the E2F is going to increase the level of the cyclin A in the cell? If you look at the promoter region of the cyclin A, that is bound by an inhibitor which is known as the cell cycle responsive element. Now, when the cell cycle responsive element is there on the promoter region of the cyclin A, you cannot actually transcribe this particular gene. Now, the E2F is going to remove this CCRE from the promoter region of the cyclin A. You can actually increase the level of the cyclin A in the cell. Now, once you have increased the level of the cyclin A in the cell, what this cyclin A is going to do then? Now, this cyclin A, as I've told you, it has the ability to bind to the CDK2 just like the cyclin E. Now, here the complex of the cyclin A and the CDK2 is known as the SCDK. So far we have discussed three types of the CDK. If you talk about the complex of the cyclin D and the CDK4, we call that as the G1 CDK. If you talk about the complex of the cyclin E to the CDK2, that was known as the G1-S CDK. And now we are having the SCDK, which is actually the complex of the cyclin A and the CDK2. Now what this complex is going to do? Now this complex is going to ensure that the gene replication goes well, the DNA replication goes well. By that I mean that you are replicating each and every nucleotide of your genome. You are not even missing a single nucleotide. Now that means that the cyclin A in the CDK2 is ensuring that the replication goes well. Another important function of this particular SCDK is it is going to ensure that the replication is only once per cell division, because if it is not once per cell division, you are going to create problems. The problem of polyploidy, for example, will be there and you will be getting abnormal kind of the cell. Now, how the cyclin A CDK2 complex or the SCDK complex is going to achieve that the DNA replication occur once per cell division? If you remember, in the G1 phase of the cell cycle, at the origin of replication, we were actually having this origin recognition complex followed by the addition of the CDC6, the CDT1 and the MCM ring, and all of them, they were actually making the pre-replicative complex, which was converted into the pre-initiation complex by the G1-S CDK. Now what the SCDK is going to do is it is going to add a phosphate group to this origin recognition complex. There are different proteins in this origin recognition complex, and the SCDK is going to phosphorylate, it is going to add phosphate group to some of the proteins. Once you have added the phosphate groups to the origin recognition complex, the CDC6 protein is actually not able to bind to this origin recognition complex in its phosphorylated form. So when the origin recognition complex is phosphorylated, its structure is not compatible with that of the CDC6. The CDC6 is not going to bind to the origin recognition complex then. Now when the CDC6 is not able to bind to the origin recognition complex, that means that is there in the cell and we do not need it anymore, because once you have filed from a particular origin, you have replicated the DNA, that means you are not interested in the function of the CDC6 now. You will need them once you want to divide the cell again. Now the SCDK is also going to phosphorylate the CDC6. Now once the CDC6 is phosphorylated, it is actually destined for ubiquitination. By that I mean that when the CDC6 got a phosphate group, another protein known as the ubiquitin is going to attach to the CDC6. And when the ubiquitin is attached to a particular protein, it is actually a death signal for that particular protein, and that protein is going to be degraded by the proteasome, and this proteasome is actually a protease complex. Now this is one of the strategy that the cell uses to remove those proteins from the cell which are not needed at that particular time. So if you look at the CDC6 at this particular time, once you have started the replication of the DNA, the replication have started from the origin and you have filed from a particular origin, you do not want to restart replication from that origin, you do not need the CDC6, you add a ubiquitin protein to the CDC6 and you are going to degrade by the help of the proteasome. What about the CDT1 then? If you remember, after the binding of the CDC6, the CDT1 protein that was going to bind to the origin recognition complex. Now once the CDC6 have been degraded, their levels are now low in the cell, the CDT1 cannot perform its function. Now the CDT1, it is not degraded, it is inhibited by another phenomena, that you are going to add an inhibitor to this CDT1, and the inhibitor of the CDT1, that is known as the geminin. So when the geminin, it binds to the CDT1, that renders the CDT1 inactive until another G1 phase. Now from where the geminin comes? Now the levels of the geminin, they are increasing in the cell because of the E2F. So the E2F is going for the transcription of the geminin. The levels of the geminin are increasing in the cell. They are going to bind to the CDT1, thereby you are rendering the CDT1 inactive. We will continue the discussion in the next video.